What sets Garvan apart from other medical research institutes is its wonderful staff and students and its leading edge technology. The most important thing for Garvan is to recruit and empower the most gifted people.
So a key highlight for me in 2016 has been the development of the Know Your Bones campaign. This is very much a collaboration between Garvan Institute and Osteoporosis Australia in what is called the Bone Alliance. We've made real progress in understanding the genetics that control our skeleton, in particular understanding how our genes might help us predict the likelihood of having a fracture in the future.
Within the Cancer Division of the Garvan, our mission is to undertake breakthrough research that not only enables discoveries of the most basic kind, but also to translate them into things that help patients in the clinic. One of the most pleasing aspects of the past year has been our step into the clinic for the first time. We've opened new clinical trials which enable the use of molecular technologies to understand each person's tumour and to personalise treatments on the basis of that information. Our researchers in breast cancer have used animal models to understand fundamental aspects of how breast cancers evolve and, more importantly, how they spread.
In 2016 we made some very significant findings that managed to be published in some very important medical journals. Paul Lee, a very talented young clinician scientist in my division, made a very important discovery showing that brown fat, when activated, can maintain control of blood sugar levels. My group, together with Kath Sudar at Victor Chang, published a very important paper showing that you have grandfather control of liver fat in an epigenetic manner.
So 2016 has been a remarkably productive year for our Division of Genomics and Epigenetics. Our division published 48 manuscripts this year, which was really an outstanding achievement. Our group participated in an international consortium collaboration run by the European Union, and here we highlighted for the first time that DNA methylation sequencing will provide a robust way of providing markers for the detection of many diseases.
It's a great time to be an immunologist. Research here at the Immunology Division really focuses on trying to understand what makes the immune system tick and how the different cells of the immune system work to protect us. Some of the major achievements from 2016 from the research done here have been making some really important insights into understanding how the immune system reacts to foreign infection versus how it reacts against its self-tissue. We've also made important inroads into understanding why individuals who have mutations in a particular gene are so susceptible to infection with some fairly common pathogens.
Application of Garvan's new genome sequencing facilities are helping us a lot. My own work has started to now look at the whole genome sequencing of bipolar patient families and investigating those early stages of trying to understand what is causing this disease, because treatments will only come once we understand their disease. John Shine, in collaboration with clinicians at St Vincent's Hospital, are investigating the cause of kidney disease, especially with familial inherited forms.
The Kinghorn Centre for Clinical Genomics has had a remarkable year. The mission of KCCG was to translate the benefits of whole genome sequencing into the clinic. 2016 was the year that happened. By launching Genome One, it became possible for families affected by genetic disease to have access to the most sophisticated genetic test in the world. Another exciting achievement this year was seeing the laboratory scale its production to more than 1,000 genomes a month. In total, the KCCG has now sequenced more than 13,000 genomes.
What drives us here at Garvan is the human impact, the ability of what we do to change lives. We do research to understand human biology and disease, but the real deliverable is to change lives. Finally, I'd like to say to everybody who supported us in 2016, thank you very much.